All right, now that we have reviewed our DADA2 results, what we're going to do is move on to the second stop in the purple line now, which is clustering sequences. So you can see now there's a green R next to alpha rarefaction, which means we can do that step now. So that is indeed the step we're going to do. So click on alpha rarefaction. And what this is doing is essentially taking a subset of reads from each of our samples, and I'll explain in the next steps the results we get and how to interpret that. But we have two different options here for inputting in different parameters. For the minimum rarefaction depth, this is basically the minimum number of reads to try taking, as 1, keep that at 1. The maximum rarefaction depth is going to be whatever we noted from that previous step in DADA2. So I wrote down 7401, that was our maximum frequency from that frequency per sample table. So that's the information we want to enter in here. So basically we're telling this step, you don't need to try sampling more than 7401 reads because none of our samples have more than that number anyways. So this is the maximum number of reads any of our samples have. We don't want to waste computing power trying to do more reads than that. So that's the only thing you have to change, and then you click Submit Job. It'll tell you it's processing, and then eventually a new page will pop up that says the job is running. And what I'm going to do is actually, so this is the screen you should see. Job is currently running. Results will be displayed here once it's finished. And that'll take a little while. Just for the sake of this video, I'm going to switch to a different project and show you what those results look like. So I've already run them. Okay. So when you have finished, when DNA Subway has finished with that alpha rarefaction job, and you can see here it has the parameters that we set. We told it to use that max depth of 7401. What we want to do is look at the results here, this alpha rarefaction plot. And what we want to do is change the metric to Shannon. So what this plot is giving us is essentially different metrics of diversity. So in ecology, maybe you've heard of these different diversity metrics. Shannon's diversity index is one way to estimate how taxonomically diverse a sample is, how many different species are there. So we're going to use that particular metric to look at our samples. Shannon is our metric. For the sample metadata column, change that to sample. And let me make this bigger. And what we have here now is this plot. So on the x-axis of our plot, sorry, I'm not sure if my head's in the way here. On the x-axis of the plot, we have the sequencing depth. So remember, we told it to do between 1 and roughly 7,000. The y-axis is that Shannon diversity index. So the higher the diversity index, the more diverse that sample is, the more bacterial taxa we identified in that sample. And each one of these differently colored lines is a different sample, so a different male or female snake. And the main thing we're looking for in this plot is where the Shannon diversity starts to plateau. So that's really, we're interested in this part of the graph here. And the reason for that is, if you look here, so some of our samples have more than 7,000 reads, but only one sample has that many. So if we tell the subsequent steps that we want to use 7,000 reads for each sample, we're only going to be including this one sample. Every other sample is going to be thrown out because they don't have that many reads. But B, the other reason why we don't want to do that is that it doesn't give us any extra information. So if we look at these plots, here at 7,000 reads, we're at about 5.5 Shannon diversity index. You get that same diversity index if you only use about 800 reads per sample. And so what we're really looking for here is kind of a Goldilocks just right sweet spot where you're using enough reads per sample to hit the maximum diversity that we're recovering in our samples while also not wasting computing time and power. So the more reads you use per sample, the more computing time and effort it takes. But in this case, it looks like because this graph plateaus at around 800 reads, that's really all the reads we need to use per sample to identify the different taxa. If we go above that, we're really not gaining any new information because, again, this graph is plateaued here. So it doesn't matter if you're using 1,000 or 3,000 reads, that line is flat. The diversity index is the same, even if we go above that. So the main thing that we're looking at here is our graph here plateaus around 800 reads. So make sure you make a note of that make a note of that number, that we want about 800 reads per sample, and then we'll use that in subsequent steps.